thanks everyone for coming. It's my pleasure to introduce PhD student and a researcher at the Genome Institute of Singapore at A-Star. And he'll be talking to us about his work on untangling. I think the work is really interesting because these are some works that are really on the cutting edge of biology. I'm really excited to find out how he's using GNNs, but also things like neural networks and neural combinatorial optimization for a really meaningful research.
Thank you very much. So, hi everyone, my name is Novo, and as Chaitanya said, I'll be talking about untangling genome assembly graphs with graph neural networks. So, I just jump straight into it, and the core of this problem is de novo genome assembly. So, what is that? Let's first imagine an illustrative example, kind of similar to de novo genome assembly, and that is, imagine you have a pile of books which are all copies of all the same book, and you push them to a shredder, like a shredding machine, and what you have at the end is lots of tiny pieces, lots of tiny shreds, which you have no idea like which part of the book they belong to. The only thing you see is the sequence. During the process, some of these tiny pieces get lost, some of them get damaged, and your goal is now to reconstruct one copy of the original book. So the only way you could do this is by aligning all these pieces together and trying to find some meaningful sentences. In de novo genome assembly, it's very similar because our goal is to reconstruct the original genomic sequence from a set of short DNA fragments. De novo here stands for anew. We have no idea what we are assembling, what the genome sequence looks like, the original one. We just have these short fragments. And as in the example with the shredder, limitations are imperfect sequencing technologies, which produce errors in the short sequences. Now that we have stated the problem, a good question also is to ask why we are doing it. So there are several fundamental aspects, things interesting from a standpoint of a fundamental biology, like exploring genetic diversity and evolution, how different species evolved over time, or how different conditions in the environment influenced their characteristics and their genomes. And from an application standpoint, we are mainly interested in the human genome. So in order to study human genetics and diseases, we would need to have a human reference genome. For this, since we don't know it inherently, we would need to construct it anew with no knowledge about it. And then we can build upon that towards some more applicable directions, such as this personalized medicine. How to get there, I'll explain shortly. But the human reference genome, we actually already have. Around 2 years ago, people from Telomere to Telomere Consortium have assembled a complete human genome, published it in a special issue of Science. And this is the first time ever that the whole genome with no bases left unplaced or no gaps in the genome has been assembled in such a way. So that was a huge thing in the community. But that does not mean that the job here is done. Why? Because this is just a single reference, which does not entirely correctly represent the entire human species, like every single person on the planet. There are inherently some biases in the genome, which then affect variant discovery and affect discovery also of the genetic diseases and accuracy of analysis. So the field is going towards assembling, not doing analysis just on this single genome that we have, but on assembling genomes of multiple people, as many as possible, in fact, of different ethnicities. That would give us a lot more comprehensive view of genetic variations within the human species. And that was, in fact, also done relatively recently. It's since less than a month ago, a draft human pangenome has been published, and it includes 47 human genomes, which have been connected in this kind of pangenome graph in such a way where you align all the genomes that have been assembled, and then you can account for differences in them. This, as I said, gives you a lot more insight into how different ethnicities, how different groups of people, how their genomes look like, and how how they would react to certain drugs. Ideally, for each patient, we could sequence their genome, get that information quickly, reliably, and cheaply, and then we would be able to tell how they would react to certain drugs and target therapies specifically for them. That's not really feasible. So when we're doing it, and probably in the future, it will be done with some other technologies, something that's cheaper and faster, because de novo genome assembly is actually computationally quite intensive and expensive. But in order to get these cheaper technologies and map them onto the reference genome, you need to have a reliable reference. And that you get by assembling genomes of different groups of people. So here you can see that in this study, there was mainly people from with African descent, and you can see dots here around the map. But now it's all about the granularity. This is still like the resolution of this is not too good. So ideally, we would want to cover this map with as many dots as possible.
Can I get a clarification?
Yeah.
By the way, are you okay to be interrupted?
Yeah, sure. Go ahead.
So from what I know of biology, and my biology is very dull, the genome between two people are like 99% identical.
Yeah.
Those 1% differs between the pairs. Like even you have different pair than you and your brother, for example. So given these reads that we have from a population, what is the target that we try to reconstruct, knowing that the 1% difference will be scattered across many different places? What is the ground truth data?
The thing, there is no ground truth per se for the new genomes that you're assembling. That's why it's called de novo. But they showed in this paper actually, or they mentioned it, that even though the variations between genomes of different humans, they are relatively small, these variations are usually so-called structural variations, which affect how genes work and like how the function of the genes, basically. So if you have a reference genome which is not reliable for you specifically, for example, the previous one that I mentioned, the human genome reference, that belonged, I think, to a white American, which probably means there are some like significant structural variations, differences in structural variations between that genome and people from African or Chinese descent. So you want to account for these differences. You want to find them. And that way, like if you are treating then people with, let's say, Chinese descent, it was a lot better to map their genomic data onto the Chinese reference genome than on the one which is of a white American. Because basically you don't even know which parts you are missing, what are the errors and what are the structural variations you have not discovered.
Thanks.
No problem. So how do we do this? The process here starts with a genome, like a biological sample that we obtain from the biologist. They multiplicate it and then they sequence it with the machines. What you get is that set of reads for which you know nothing about. It's just a large amount of sequences with no ordering retained between them. And then your task is to find the ordering. You overlap all the sequences, you take one, you overlap it against all the other sequences. And from this information, you can build the graph. With this, we call an assembly graph. And each node in the graph represents a sequence, and a suffix prefix overlap represents an edge in the graph. So the next step here, what most of the standard assemblers do, is called layout. And they try to find some patterns in the graph and simplify them in a way that they remove nodes and edges to get some paths in the graph. They basically simplify the graph into the paths by removing nodes and edges. And these paths can then be translated into genomic sequences, and that is your assembly genomes, which you can then, if you have a reference genome, you can compare it with your ground truth. If not, this is the best you can get. There are some additional techniques that try to bridge these gaps, but in some cases, this is what you have. The problem here is that there are some patterns which you can recognize and simplify, but there are some which you have no idea what to do with. And then standard algorithms, when they find themselves in the situations where they don't know what to do, they simply cut this into pieces, they cut out parts of the graph, and you are left with gaps. And in our approach, the premise is that we want to find a path directly through the graph instead of removing nodes and edges in order to get a more contiguous or a less fragmented assembly. So instead of cutting out parts, we want to untangle them. That is, find some path that is correct and that leads to less fragments. What is the problem here? The problem is basically that the graphs can be huge. This is a graph of human chromosome 20, which is one of the smallest and the simplest chromosomes. And it still looks pretty large and untangled. And it's very difficult to find some patterns for which you can construct rules. In case of this, do it a certain way. How do assemblers do it? Like, there are some structures which are known. Yeah, sure.
Sorry, for that example that you're giving, so that's for one person. So this is still not for the pangenome. It's like
okay. In this talk, I will mainly be focusing on one person. For the pangenome, that was more of a motivation. Like, we have a single human genome. That doesn't necessarily mean that we don't have to sequence other human genomes. Like, people are actively working in the field on sequencing more and more human genomes, and that's like the result of this is the draft pan genome, which was published a month ago.
Okay.
But yeah, this is a single chromosome of one person, and it's pretty messy. So there are some structures in the graph which are easy to identify and which assemblers traditionally remove. So these are transitive edges, such as these structures here. These are dead ends, and these so-called bubbles, which often stem from some single nucleotide polymorphisms or structural variations. But then there are also tangles in the graph, as I mentioned, which we don't know how to solve. More on that slightly later. And regarding this was our initial pilot study done with Petra Veličković from DeepMind, where we basically wanted to see whether graph neural networks can simulate and whether they can recognize and also simplify the graph in a correct way, whether it can solve it in such a way that resembles, at least to a certain extent, the work done by algorithms and in what time. The times were comparable, and the results were as well, at least on the small graphs that I tested, but it was done on synthetic data. And as I said, this was just a pilot study. After this, we wanted to take the same approach to simplifying the full larger real human genomes. However, that proved to be quite difficult, so we took a slightly different approach. As for the next steps that the assemblers are doing, there is no cookbook for this. Like, every assembler does something different. One assembler implemented by a former colleague from my lab, his method was to plot the assembly graph onto a 2D plane, identify these long edges, and then cut them out. Why it's difficult to say, but it worked fairly well. The idea actually was that if you have these long edges, you would expect them that if the graph is plotted correctly or in a certain way, it would connect the distant genomic regions. It's a heuristic. It worked in some cases, in some not, but overall the performance was not bad. But this is an illustration of how every assembler will take its own path towards simplifying the graph. The only like proper way of doing it, of simplifying the graph without losing information, is to do it manually. And this is exactly how the people from Telomere to Telomere Consortium have assembled the full human genome with no gaps. They used different types of data, some of them shorter but more accurate, some of them longer but less accurate. And they had to not cut the tangles, but then like use these longer reads to find paths directly through these tangles. That required a lot of work from many researchers, bioinformaticians, and it was extensive work that took like several months. Yeah.
Did people also manually do some of these tasks? Like at the start of the Human Genome Project, was some of it like literally manual work as well?
Well, not really at the start, it was more like a final phase. Like they got as close as they could to resolving it automatically with the assemblers. But then when you, the main problem here were the centromeres, which are known for having like large repetitive regions where basically it seems like everything is connected with everything. And then you don't know how long these repetitive regions are, for example. So what you do is you use these like longer reads that you can just kind of guide through the tangles. But it's also like it requires some manual work. And then yes, and for CHM13, the initial human reference, and for the pen genome, I think there as well they used significant amount of manual labor. The drawbacks of such approaches is that the heuristics used, they very frequently rely on some handpicked parameters. By handpicked, I mean they are evaluated on a few kind of more popular genomes, such as is human or E. coli, and there is no real guarantee that people work on genomes of other species that you are assembling. So you would want to, if you would want to find a better set of parameters for new genome, you couldn't really tweak them slightly and expect to get better results. You might need to start all over again. We wanted to implement a method that would find the patterns directly from the data, not do all the manual inspection of the complex regions.
Yeah, so because
And so that it scales to new genomes in a way that if you have that, yeah, it automatically can learn from having new genomes assembled. And that basically means use machine learning. The more data you have, like if the current model works fine on the existing data and you get some new genomes that doesn't perform well, you can train more data, the more data you have. And since we are dealing with graphs, I've been mostly talking about graphs now. I will also, it is fairly logical that we are going to talk about graph neural networks now. The pipeline here is to first define the objective. So far, it was very vague. I just said that we want a less fragmented genome and that we were going to use graph neural networks for this. But how exactly? Once we do that, we have to define a training set. And since we're doing deep learning, we usually need a lot of training data. So we're going to use a simulator for which we can simulate large amount of data, and we can also get the positions from which we can compute the labels. And then after this is done, we can simply define the model and train it on the stated objective. In inference, we use the trained model to get predictions. Those will be certain probabilities, as you will see. And from predictions, we want to get context. Those are the sequences in the genome. So how do we do that, and how do we evaluate the final quality of the genome? Objective is, as I said, unfragmented assembly. And instead of cutting the regions from the graph, we want to find a path through it. That basically means that we need to know which edges we can traverse and which we should avoid. So that comes down to edge classification. We want to know which edges are good, which are bad. And the whole problem can be stated in the end as edge classification. And for this, we use like binary cross-entropy loss, and then we need lots of labeled training data.
Can I get
Yeah.
small clarification? If the task to find any path, and if it's any path, could there be more than one possible path?
Any correct path. Let's say this way. there is not necessarily a single good path that leads to the optimal genome, but just because it comes from the start until the end doesn't necessarily mean it's a good path. You could have some loop in your assembly graph.
Okay, path without loop.
Yeah.
And is it necessarily unique?
No, because of the structures such as transitive edges, and so you can take some different bubbles as well. You can take different paths and end at the same assembly.
Okay, in the end, probably generate the training data so that you have a unique label to train towards.
We're gonna get here
Okay.
now. By resampling the reference, basically. So I use a simulator which can simulate the data so that it resembles the real data used in the in assembly. And every time I resample the reference, it different trees with different lengths and different error distributions come from different parts of the genome. That influences differences in the assembly graphs significantly enough so that each graph can be treated as a different training example, at least until a certain point. I'm not saying you can do like infinitely many training examples from a single chromosome like this, but we tested and like it works fine until a certain threshold. Because we are simulating it, we can also get positional information for each read. We know exactly where it comes from, which starting and end position on the genome it comes from, and then with an algorithm you can compute whether an edge is good or not, whether it leads to the optimal solution or not. One caveat here is that there could be a difference between actual between the simulated reads and the actual read error profile. This is one of the, in my opinion, best simulators out there for the hi-fi data that we are using, but there still are some differences in the simulated data and the real data. And this can cause some problems down the line, but it's pretty difficult to fix. So there is a certain level of abstraction, and yeah, we have to accept this. After this, we generate the graph, and in theory, we could use any assembler that follows this paradigm of constructing assembly graphs from the sequences. We take the graphs from the and we take the graphs from the overlap phase, which I stated earlier, without any simplifications done on them. So we take each graph contains all the information about the overlaps, no edges removed. And then we leverage those positions that we saved during the read simulation phase to construct the labels, which I said it's only possible with the simulated data, but the reads, even simulated from the same sequences, same chromosomes, they will produce somewhat different graphs so that like you can train on more instances simulated from the same chromosome. That's something that's fairly interesting, actually, as you'll see later. And yes, for this, we'll use graph neural networks. Most of you here are very familiar with graph neural networks, so I will not spend a lot of time here. But the idea is that, yes, like you want to iteratively update the state of a certain node. So we do message passing from all its neighbors towards this node that we want to update, whose representation we want to update. And then we aggregate the messages and push it to another update function, and we get the new state. That GNN, that is the backbone of our model. Our input is graph, which has certain node and edge features. Edge features here are overlap length and overlap similarity. So the length of two reads and the similarity between the sequence of one read and another, which is overlap. Node features currently in degree and out degree of each node. There was a lot of experimentation here. We tried different positional encodings. We tried several tricks. There's also always like kind of on par. This seemed to work slightly better, so we stuck with it. But it's I cannot give you like a definite reason why this is good and other things are not. It's the results are fairly similar. And which particular model we will use, which particular GNN layer, we opted for gated GCN for several reasons. One is that in the benchmarking paper, GNN performed well over several different tasks. This is for the edge classification task, where it achieved the best results compared to other models. And another reason is that it was used in Chaitanya's work on traveling salesman problem, where they achieved the state-of-the-art results with this model. Traveling salesman problem is, in fact, pretty similar to what we are doing here. You also start with the graph. You also want to predict which edges lead you to the optimal solution. And from this, you get certain probabilities. And then you have to decode this with some search algorithm. Chaitanya used beam search. I tried beam search, but due to some technical limitations, we are going to use greedy. However, there is one slight difference. We have to modify this gated GCN. And the main reason here is that the graphs done in TSP, used in TSP, they are undirected. And we use directed graphs. Directed basically here means which edge, which read is overlapped with suffix and which with a prefix. So this is important information in assembly graphs. It gives a kind of inherent directionality, and like what is the beginning, what is the end of the graph. And from this, you can decode like what is the beginning and end of the genome. So we actually had to modify this network so that we independently treat incoming edges and outgoing edges. Basically, if you compare it with previous, we just had to do this part twice, where you do message aggregation from neighbors. You just split it into two pieces, here and here. Yeah.
Do you learn different sets of weights for incoming and outgoing edges as well?
Yeah. Yes. So matrices here are A2 and A3, meaning we completely split these two.
Can I add something?
Yeah.
This is actually true with the more expressive than vanilla GNN. There was recently a paper, I'll get the name wrong.
Yes, this one, right?
This one.
Yeah.
This one. I found this very interesting.
Yeah.
Because this is something that to us seemed like obvious thing. You cannot treat your directed graphs as undirected. So we just did the thing ourselves, and it kind of it produced significantly better results. But it was nice to see that somebody took this and like mathematically, formally explained what's going on, and they provided a framework for treating directed graphs with, I guess, directed versions of the GNN models. Dobrik, another question, or is this?
You know, it's good that I can read your thoughts.
Yeah, or I can read yours. I put them at the sides first. But yes, the idea is exactly the same as what we did about a year and a half ago, to separate aggregations of the incoming and outgoing edges. There are different things, simply treat them differently, have like split equations into two parts. Okay, so training is a usual training setup where we push the graph into the GNN model. We calculate some probabilities, we compare them with the labels, compute the loss, and update the weights. There is a certain problem here. As I said, the graphs are huge. And if the model is slightly bigger, or you cannot put the whole thing into your GPU memory. If you don't use GPU, it takes forever. So we had to use another trick here. We had to partition our graphs into smaller subgraphs. For this, we used Metis. This is a really fast algorithm that partitions your graph in a certain number of partitions with the idea of cutting the minimal amount of edges in the graph. One thing not really mentioned here is that it can also be useful to include one hop neighborhood. So especially here, we are here doing edge classification on the graphs. So if you partition the graph in such a way, you are losing information. You are cutting out edges, which could potentially be very important. In genome assembly, there's a problem as you may see later, that there is a big discrepancy between the task that I'm training on, which is edge classification, and the final evaluation, meaning how good our assembly is. You can have one error in the graph, and that can break your assembly into fragments, because it's not very reactive, or it's very reactive to it. Minor changes in the probabilities in the predictions can influence how the greedy algorithm decodes the graphs and how it creates the genome assembly. So we actually also include one hop neighborhood from each of these, and then like we cover these edges which are here missing. We actually cover each edge twice. In inference, no such problem. In inference, if we would, we just do it on CPU, because we haven't come with a smart way of partitioning the graph and then getting unambiguous probabilities for these edges that would be cut with Metis. And also in inference, it's relatively fast. This is not the bottleneck of genome assembly. It's the overlap phase where you have to take all the reads and overlap them with all the other reads. So even if it takes a few more minutes than it should, it's okay. And we finally come to the encoding. We start from a high probability edge, or actually like we sample multiple high probability edges with the Bernoulli sample. For each edge, we take its source and destination nodes, and we just do greedy decoding forwards and backwards. What you end up with are these walks, and each walk represents a single genomic fragment. You repeat this until there's no more nodes in the graph, or until a certain threshold that the graph is too small, or your decoded walks are too small. You stop, you get a collection of walks, and then you need to translate them into sequences, which is a straightforward thing. Since each node is represented with a string, you just find these overlapping regions, and that's it. You concatenate it in such a way that you take into account the overlaps. And the result of this is the assembled genome. The final output is the same as is for all the standard genome assemblers. So how do we evaluate it? Well, since the output is the same, we can also compare it against all the other standard metrics. Number of contigs, meaning number of fragments in the genome, the length of the longest contig. Genome fraction is what's the portion of the genome that we have covered with our assembly. NG50 and NGA50, which are the most indicative, perhaps, and also the most difficult ones to explain. So we'll start with this. In a way, they're both kind of the medians length of the contig, but NG50 is like you sort your contigs by length from longest. Okay, these are the contigs. This is the genome assembly, your assembly, and this is the reference genome. You sort them from longest until the shortest, and then you take the contig, the length of the contig, with which you pass 50% of your reference genome. So 50% of 500 is 250. There is 120 plus 80 is 200. You need one more plus 70, 270, and that's more than 250. So NG50 of this would be 70. It's a bit convoluted. Don't ask me why. It's the way it is. But in a way, it kind of gives you the median value of your assembly contigs of their lengths. NGA50 is similar, but it additionally maps the assembled contigs onto the reference genome. So for example, if some contig has been disassembled, that it seems to be 120 base pairs long, but in fact, it comes from two different regions. It will map it to the reference genome, and it will break it into two pieces. So you no longer have 120, you have 2 times 60. And then you repeat the process again. And this time, the contig that covers 50% is no longer the one with the length 70, Now is the one with the length 60. And finally, we are getting to the final stage, which are the results. So we start with a very simple setting where we have trained on simulated data, errorless, perfect reads with no errors. We used Raven assembler simply because it was easy to manipulate it to behave like we wanted, because it was implemented by the colleague from the lab. And we evaluated also simulated hi-fi reads of other human chromosomes. So for this, we used 15 times chromosome 19, and then we wanted to evaluate on everything else. You can notice a few things here. First, we assembled in a lot less. I'll assemble it here. You don't see because of weight.
It's the one on the left is yours.
Yeah, the one on the left is mine. I'm just trying to. I only now noticed.
This might still screen sharing.
Okay,
It's
you
clear.
cannot see, but this says
Yeah,
mine.
yeah,
This says not mine.
It's clear.
Yeah. GNN assembles in a lot less context than the standard assembler that we compared to. And we are comparing with this one because we start from the same graphs. We start from the same, if we start from the same graph, then we are only comparing the layout phase, the graph simplification phase versus the GNN. Less context, our contexts are a lot longer in a similar or a slightly better genome fraction, like the amount of genome that we cover. And in some cases, NG and NG50 are somewhat comparable, but in some cases, it's a lot longer in the case of GNN. This is the result we were aiming for, and this is exactly what pushed us to do additional tests on real data. Before that, we actually wanted to do a sanity check, whether GNNs are even learning anything, whether it's something trivial. Maybe the overlap lengths or the overlap similarities are indicative enough of good assembly that you can simply follow them. Yeah.
During the previous table, so you train on data only from chromosome 19.
Yeah.
Is that, but so when you evaluate the chromosome 19, are you evaluating on the training data?
No. So this is like 15 times like different instances obtained with different sampling of chromosome 19. And I evaluated on another instance. It is kind of shady. That's why there's a start here. And you can just like neglect this row.
Yes.
So we tried to be transparent, but kind of for the sake of completeness, we included chromosome 19 as well, even though, yeah, this is maybe not the best practice.
But is Raven the sort of state-of-the-art genome assembler?
There's a thing it's kind of state-of-the-art for a different type of reads, for Oxford nanopore reads. This was done with PacBio hi-fi reads. So we chose Raven for two reasons. One, it was implemented in our lab, and then when we needed some modifications to how to produce the assembly graphs that we needed, it was easy to modify it. Second was it worked fairly well with different types of data. A lot of assemblers are tailored for one specific type of data, either for Oxford nanopore long reads or for PacBio, which are somewhat shorter. And Raven performs fairly well. But interesting that you say this was one of the main concerns of the reviewers whenever we were submitting the paper, and I will address this soon. Yeah.
Coming back to my question,
Yeah.
if I understand correctly, this table, the metrics that you get for chromosome 19 sometimes are not as good as other chromosomes, even though it was trained on 19.
Yeah.
Any intuition why, any reason why you think that might happen, or?
Sure, you know. Sure, you know. That's that's one of the pitfalls in this problem. It's this discrepancy, right? I can get some accuracy in the predictions on the edges, what's good, what's not. I can try to reduce false positive, false negative rates. I found those metrics to be kind of indicative of performance. But then when you run the decoding and when you start actually comparing with the on the genome level, things happen, right? And it's really difficult to then transfer from this to analyze what went wrong in the genome in the predictions. I try to do it. In certain cases, it helped me a lot. I recognize which situations make assembly worse or better. But I was not always able to do it.
Can I try to make a guess?
Yeah.
Do you use the same number of layers across all chromosomes? Or is it like an RNN that repeats for a different number of steps?
It's the same number of layers. So I would treat it like, okay, like this network is gonna have.
So could it be that, let's say, chromosome 1 is so much shorter than the chromosome 19?
Actually, chromosome 19 is one of the shortest ones.
I don't know biology.
Okay, no, so this is one of the shortest ones.
Yeah, okay, so could these results be due to the number of layers?
They could be, but then that would mean that your network with the fixed number of layers is trained on graphs of certain size, and then it kind of performs better on larger genomes, on larger graphs, than on the ones that you were trained on. It's
Yeah, to me, this is a little bit the kind of shady parts. Because if you consider that the network knows that's some heuristic internally, the time you need to execute this heuristic should change with the graph with the graph size.
yeah,
Correct?
yeah.
And now here we use the same number of layers.
Yeah.
Meaning we do the same number of iterations, so the things that come better on larger graphs, which is a little bit counterintuitive.
I think it's very counterintuitive, but it might be, as we discussed before, due to not just the network, but the robustness of the decoding part. Since in the decoding, as I said, I don't just do it once. I don't just take one edge and decode from it. I sample certain number, in my case 100, and I find 100 candidate walks and then take the best one of those. The best one in this context means the longest one. And then I repeat this. In every iteration, I sample 100 edges and do the decoding from each.
Which chromosomes are the smaller ones, the latter or the first ones?
These here. So
Could it be the case that the sampling for the smaller chromosomes leads to less diversity in the sample deletions?
it could mean.
Yeah.
I guess it could mean.
And this could be like a possible explanation why I get really good results in large graphs, because you get a really diverse set of solutions. Anyway.
It's possible. It's possible. Yeah. Yeah, it's very possible. It's but it's difficult to say why. So we then did a sanity check where we just wanted to check if from the same edges that we sampled in the previous table, in the previous table, right, I would sample certain edges and then I'd run decoding from those following the probabilities. What if from the same edges we just follow either the longest overlap or the longest similarity, or the best high similarity? You would expect that the higher similarity between two sequences, the better overlap is and more chance that it is the correct one. Similar with the length. If the sequences if there's a longer overlap between them, higher chance that the overlap in fact really is good. So it performed so bad that in some cases, not even 50% of the genome was assembled. So that's why there is no reported value for NG and NG50. So the network probably did learn something. And then we tested it on real data. And still, in a less context with usually longest context and similar comparable genome fraction, we can in certain cases get significantly longer NG and NG50. And I will talk about this more soon, but you can notice that in some cases there is also a big discrepancy between NG and NG50 here. So that basically stands for like it decodes longer sequences, but in some cases they are wrong. In some cases they're still better than the ones provided here with the heuristics from Raven, but ideally you want these to be close. And as the last thing here, we notice that also per base errors, the mismatches in the bases and the insertions and deletions were lower. So the questions here is, yes, like this NG50 longer than NGA50, I call it more aggressive decoding, which is the whole idea in a way of our approach, that when there is ambiguity, when you don't know what to do in a certain complex region, you try to untangle it instead of cutting it out. But then you can assemble it incorrectly. So we try to fix this. And then the questions raised also before, can this generalize to whole genomes? All this was done only on individual chromosomes. Can it generalize to other species? And can it really be used with any other assembler? I said, like, if we have, this method is completely agnostic with respect to the assembler. So in theory, at least, if you construct an assembly graph with a different assembler, not Raven, but something else, and use our method on these graphs, such as is a method that really is state of the art for these type of reads, would it also perform well? Well, let's scale it first to whole genomes. We still use Raven, and one thing also that we fixed is I included more errors. This is not longer trained on perfect sequences. I started using a different simulator, which includes, which makes a lot more reliable data, and this helped a lot. Surprise, surprise. But it was important to have reads that are similar to the real case scenario. And we do manage to, when scaling on full graphs, we still get higher, like better contiguity, less fragments, and even the NG and NG50 are fairly close. Now there is, the discrepancy is a lot less than it was in some cases before. Can we use it with different assembler? HiFASM is the state-of-the-art assembler for hi-fi reads. It was the one predominantly used, I think, in the assembling of human pangenome draft. For all those 47 assemblers, they were predominantly using HiFASM, and then doing manual labor of untangling and doing trio binning with different types of data and many, many tricks. So can we beat HiFASM if we take its graphs and instead of using its heuristics, we just plug in GNN? I'm glancing over a lot here. This was really difficult to achieve. This took almost a year. But we showed that, like, we can, in many cases, get comparable results, and in some cases, we managed to get even better results than HiFASM. So this NG and NG50 of 111 megabytes first, the best of my knowledge, that's like the state-of-the-art results for the genome assembly, human genome assembly.
I hope I'm not getting annoying. But
No, go for it.
For those not familiar with the topic, what does it mean, like, to combine, let's say, HiFASM with GNN? Like, which parts are HiFASM, which parts GNN?
HiFASM is the graph construction part, where you take the reads, the sequences, you overlap them, and the output of that phase is an assembly graph.
Yeah, fair.
I take this, and then I use GNN to get the probabilities.
Okay, and if we don't use the GNN, what's the difference?
The difference is that then, instead of using GNN, you use the HiFASM's heuristics for the layout phase for graph simplifications, which are those transitive edges and bubbles and other things that are done. I'm not even sure which all the steps are. So yeah, the whole idea is that instead of using heuristics, we plug in a GNN. And yes, like, we managed to get better results with the GNN. And also know this was trained on human data, but it was not trained on the same type of human data as is shown here. It's two different human genomes. And these we still train on simulated reads, so we don't use real reads in training at all. But if that's not enough, okay, can we then generalize to different species? For this, we use yeast. Why yeast? Because so far we have the human genome that we have, that I have shown here, that I have talked about the results on, is haploid. That basically means that instead of having like 23 pairs of chromosomes, it only has 23 chromosomes. That means it only has the genetic information effectively of a single parent. Whereas usually people have parental and paternal and maternal genetic information. Here you only have half. It's significantly easier to work with haploid genomes and to assemble those. So for the first step, we took this as a task. So that's one of the limitations, as I will explain later. And also this yeast is also haploid, so we evaluate on this. And still with Raven and with high-fi reads, we again managed to achieve somewhat higher NG and NGA50 with the same graph fraction. And I stopped reporting here, but in less context. And yes, the limitations of this work are that we start with the graphs produced by different assemblers. We cannot fix the errors that already exist in the graphs. And this graph construction phase, it's also heavily based on heuristics and approximations. If it would be done in an exact manner, it would take too long. It would literally take 2 years to align all the sequences. The second one is that we are currently unable to do phased assembly in any way, whereas HiFiSM can do that fairly well. It can just on its own produce phased assemblies, meaning 2 split haploids to a certain extent. But if you provide additional data, such as parental data or Hi-C data, it works even better, which our method cannot do yet. And the last thing is there's actually a lack of high quality genomes of other species, or until recently, there was a lack of high quality species of human even. So we had a very limited training set. We had very limited set of things that are labeled sufficiently enough that we can train on them. And then it was very difficult also to generalize to any other genomes. But in conclusion, this is an entirely novel approach to resolving assembly graphs. Again, to the best of my knowledge, nobody tried anything related to graph neural networks and to genome assembly. These are still preliminary results, which indicate improvements over existing methods. And ability to generalize the assembly graphs of different assemblers is, I find it quite remarkable, even though I cannot fully explain why it generalizes so well, why I am able to train on certain types of graphs, and then it generalizes to something that seems to be entirely different. It is kind of counterintuitive. It could be that it's just the robustness of not the GNN, but of the entire pipeline. So that's also something that should be analyzed in a lot more depth. And finally, like with more telomere to telomere genomes assembled, the method should get even more robust. It should, in theory, get more powerful because we will have more training data. And the more different genomes we see in training, the more different species, the more different types of reads, the GNN should be able to generalize better to even new unseen genomes. So for this, I would like to thank to all my collaborators, Xavier Bresson, Thomas Laurent, Martin Schmitz, and Mile Šikić. And thank you everyone for your attention. There was a lot of questions during the talk. If anybody has any questions now, please feel free. There are a few questions online. So,
but before that, we can also stop the recording so that if people want to ask controversial questions,
sure,
they can do that.
Okay.
I'll do that. Yeah.